All right, so we finished chapter 2 talking about the properties of water and the chemical bonds that form between different elements because of the way that the electrons interact with each other outside in that valence electron shell. Today, we're going to dig a little bit more into what some people would call biomolecules or macromolecules or organic molecules. If you've ever tracked diet stuff, they might be like, oh, you're tracking your macros. What they're really talking about are the molecules that are found in all cells that are making up those component parts and allowing cells to function in their normal activities. Those are made of elements that are linked together through either ionic or covalent bonds, and we're going to dig into it a little bit more. So let's talk about what you should be doing in note-taking for this particular chapter. There is a biomolecules table in chapter 3 in Canvas, and it's a very simple table. It's just a Word document, and what that table is doing is it's allowing you to track the differences between carbs, lipids, proteins, and nucleic acids from this particular lecture. I would strongly suggest having those and either having it printed off or making a version of yourself on a piece of paper or having it open on another screen and pausing and filling it in when we cover one of those macromolecules. So make sure that you can do that. That is going to be fulfilling your chapter objectives for this particular chapter. Okay, so let's start off by identifying the differences between organic and inorganic molecules. So sometimes people get this wrong idea that you as a living organism do not need inorganic molecules, which is absolutely not true, because inorganic molecules are things like water and salt. You absolutely need those, and as we learned in chapter 2, without water you will die within several days. So it's not a good thing to say you don't need that. Instead, organic and inorganic molecules are differentiated by the presence of a carbon and hydrogen chain or the absence of that chain. So organic molecules have carbon and hydrogen, which is why we are carbon-based organic life forms, because we are mostly made of carbon and hydrogen and oxygen because of water. And then inorganic molecules don't have those. Now, you still need a lot of those for daily functionality and like well-being. I know we talked a little bit about how some of those elements that we don't see as the big component parts of a living organism are still really important. Things like iron and magnesium, iodine, those sorts of things keep you healthy. But in reality, when we're talking about our macromolecules or biomolecules, what we are talking about is organic molecules. Now, if you remember, carbon is absolutely fantastic. So carbon is 6 on the periodic table of elements, which means that it has 6 electrons, okay? And if you remember the octet rule, an atom wants to have 8 electrons in its outermost valence shell. So carbon has 4 electrons in its outermost shell, and as such, it can share electrons with up to 4 other elements. Now, carbon doesn't like to donate or receive electrons. It doesn't like to form ionic bonds. It always forms covalent bonds, which can be either polar or non-polar covalent bonds. Okay, carbon can bond with as many as 4 other elements, including other carbons, and it can form double and triple covalent bonds as well. Generally speaking, carbon is always going to be holding on to a hydrogen. That's just general rule of thumb, especially if we're talking about organic molecules. And so you know right away that it doesn't matter which of the macromolecules we're talking about, they are going to have carbon and hydrogen involved in that chemical composition of that particular molecule. Okay, so here is your first check your understanding. Make sure you can answer this, and once you're done answering it, come back and continue your lecture. All right, let's talk about these biomolecules, okay? I like to abbreviate it as biomolecules. For me, it makes sense because we're talking about the component parts of cells and organisms. And there are 4 major categories that we look at in biology: carbohydrates, lipids, proteins, and nucleic acids. Okay, your body needs these in various ways, shapes, and forms that are going to help you maintain your functionality and well-being. And your cells are made of all of these components. In fact, all cells are made of these components. It doesn't matter if you're a bacteria cell or a blue whale cell, we are going to find carbs, lipids, proteins, and nucleic acids in those cells. So these are conserved across life and therefore are incredibly important as that building block, that building material. Okay? What's really cool about biomolecules is they work like, I like to think of them like those 3-in-1 Lego kits, where if you get one of those Lego kits, you can kind of break it down and then build it up into something, and then break it down and building up into something. You don't have to add or remove any of the Legos from the kit. They're just rearranging them. Generally speaking, these biomolecules work in the same way. Your body doesn't just take the protein from a chicken cutlet and just shove it straight into your muscle. Instead, that protein is going to be rearranged in some way, shape, or form. And this all plays into that nutrient cycle that I talked about in energy processing characteristic of life. Digestion is going to be the process that breaks these molecules into their subunits, or what we call monomers, a single unit, and then build them back into the macromolecules, biomolecules, polymers. They have a lot of kind of names, but the polymers are the larger units of these biological molecules. Okay, so let's talk about how digestion works in a little bit more detail. There are two things that are going to happen with these biomolecules. They can either be built into a large molecule, so you can take the monomer and build a polymer, or you can take that polymer and make it into monomers. So, for instance, okay, you are going to be eating, maybe you're doing workouts, and after workouts, like we like to do this at my house, you have an amino acid recovery drink. Okay, those amino acids are the building blocks of proteins. They are the monomers of that protein. So your body will take those monomers, those amino acids, and use dehydration synthesis to join those monomers by removing water molecules. Okay, so what's essentially happening is each of these monomers is going to have a hydroxide group on one end and a hydroxide group on the other end. And so as they're kind of coming together, dehydration synthesis basically says, okay, I'm going to snip out an oxygen and two hydrogens. So we take out that oxygen and two hydrogens, and that makes a water molecule, right? H2O. Your body can then use that water elsewhere, or you can release it through water vapor or sweat or whatever it may be. That oxygen on the second monomer does not like to be by itself, because now the electron it was sharing with the hydrogen is gone. It's with that hydrogen and that other oxygen. And so to keep that octet rule fulfilled, that oxygen is going to end up becoming covalently bonded to the other monomer. And in doing this, we are linking together monomers into a polymer. So we are putting amino acids together to make a protein, as an example. This happens for anything that has monomers and polymers, okay? That's awesome, and you're going to do that if you're building muscle or that sort of thing. But what if you're eating for energy, or you're eating a protein, you're eating like a chicken breast, but your body isn't going to take the chicken breast again and put it right into your muscle, right into your bicep. It's going to need to take that protein and break it down into the monomers. It does that by hydrolysis reaction, okay? Before we get into hydrolysis, let's break down the word hydrolysis, okay? Hydro means water. We're pretty familiar with this. I think most of us understand that hydro means water. Great. Lysis literally means to break. So hydrolysis reaction means to break with water. And so what it's doing is when your body has water in it, as you need, right? And you eat something, your body will utilize that water to reverse the process of building a polymer. And so you take that polymer and you break it down into its monomers by adding the water that adds an OH group to one monomer and a hydrogen to the oxygen that needs to have its octet rule fulfilled. And now you end up with those individual monomers. Okay, so for instance, this could be eating a chicken breast and breaking it into the amino acids, or having starch and having starch broken down into glucose, okay? When we see these processes, our body can then rearrange these monomers. We're not removing the water, right? We're not really removing the water. We're just rearranging it. And this is how we see those chemical reactions, those equations form, and we can kind of go back and forth. And this plays a big role in that nutrient cycle. I had said in, oh gosh, forever ago now, in chapter 1, it feels like, at least. I know it's only been 3 lectures. But you are made of carbon that has always existed. Okay, we are made of carbon and hydrogen and oxygen that have always existed. And so this is part of how that process works. Okay, so that is digestion. Now, let's dig into each of our macromolecules in a little more detail. Okay, first, we're going to talk about carbohydrates. And the cool thing about carbohydrates is they tell us what they're made of. So carbo, carbon, and then hydrate, water. We know that carbohydrates are carbon, hydrogen, and oxygen. So the elements found in a carbohydrate is carbon, hydrogen, and oxygen. Okay? And carbohydrates are a polar covalent molecule. Okay? As a polar covalent molecule, carbohydrates are hydrophilic or like water. It's why if you leave pasta in water, even if it's not boiling, that pasta will eventually absorb that water and will start to break down because carbohydrates are interacting with those water molecules. Okay? As such, carbohydrates are generally a great use for energy source. Depending on what organism you're looking at, it could be long-term or short-term energy source, but carbohydrates are absolutely useful. Okay? And that's why when you think of carbs, you tend to think of your complex carbohydrates, your breads, your potatoes, your rice, your pasta. However, carbohydrates can be your simple sugars or your complex carbohydrates. So they're classified as either monosaccharides, one sugar, mono meaning one, saccharide meaning sugar, or polysaccharides, many sugars, poly meaning many. Now, we're going to talk about both monosaccharides and polysaccharides because your monosaccharides are the monomer and your polysaccharides are the polymer of your carbohydrates. Okay, so if you're following along on your table, you should be able to fill out the monomer, the polymer, examples, and the elements found in carbohydrates, and make a note to yourself that carbohydrates are polar and therefore hydrophilic, because remember, water interacts with polar and ionic molecules. Okay, so let's talk about our monosaccharides. Again, mono means one, and saccharide means sugar. And so these are your simple sugars, your single sugars, okay? These are going to be used extensively by cellular respiration. Specifically, glucose is the one we talk about the most. And so we're going to talk about glucose in Unit 2 and talking about how it is made and how it is broken down. But we use these simple sugars as a really quick energy source. Now, you might notice that the examples I have up here are glucose, fructose, and galactose. And so you might be noticing that at the end of each of these words is the letters OSE. Okay? If you see OSE at the end of a word, that means that it is a sugar. It is a carbohydrate. Okay? And in this case, things like molasses, cherries, and yogurt are going to be a source of simple sugars and a source of that quick energy source, which, now that I'm an adult, makes sense to me, right? When I was younger and I was hungry, between meals, my mom would have me have fruit or a yogurt. And I was always kind of like, ugh, fruit or yogurt. Now, I love fruit. We just bought way too much fruit yesterday. That will get eaten. It will all get eaten. It's such a good energy source. It's got some fibers and things that are going to help you feel full. It's got vitamins in there for you. But then in addition, that fructose or that galactose is an energy source that is going to help you keep going because your body can use it almost immediately once that digestion process has started. It can take that glucose in. It doesn't have to break anything down necessarily, and glucose can go right into your bloodstream and go to your cells to be used by cellular respiration. Okay, that's really awesome. Monosaccharides, fantastic. Polysaccharides are a little bit more complex, which is why we call them your complex carbohydrates. Okay, some polysaccharides do function as energy storage molecules, things such as starch or glycogen. And then some function as structural components, such as cellulose and chitin. I know it looks like chitin, it's not, it's chitin. Okay, so I'm going to show you a picture and talk about each of these a little bit more. So these are your polymers of your monosaccharides, and these are some examples of those polymers. And let's talk about starch and glycogen first. So in your book, they have these images of a starch granule in a potato cell and glycogen in a liver cell. And really, they don't look that different. Okay, starch is highly branched, which means that it's relatively easy to break down. In fact, if you hold starch like a saltine cracker in your mouth, you will start that digestive process right away, and you'll start to break that down with an enzyme called amylase. Starch is broken down into glucose, which can then be quickly used by your body. But if you are having a big pasta dinner and then you go and sit on the couch and play video games for the next 24 hours, okay, your body isn't going to necessarily use all of that starch, all of that glucose that came from that starch right away. Instead, that starch is going to get stored in your liver as glycogen. Okay, glycogen is very similar to starch. It's still highly branched. It's still easily broken down, and the body can use it fairly quickly. Okay, now, what's really cool, sorry, I just had to stop and think about the way I was going with this. Let's talk about potatoes first. So if you've ever had a potato that you left like in the fridge too long or in a cupboard too long and it started sprouting new growth, okay, because potatoes can grow from those eyes on them. Potatoes are a way for plants to store starch as an energy source over winter and other long periods when they might not have that readily available energy. If your potato starts to sprout, it probably went from being nice and firm and wet and like hard to soft and squishy and maybe even a little bit dried up, because that potato is going through the hydrolysis reaction to break that starch into glucose that is then allowing that plant to start growing, because plants need energy to grow. Isn't that awesome? In our own bodies in the same way then, glycogen can be used as an energy source, and it's used a lot by athletes. So if you were an athlete or you know anyone who was an athlete and they would do the big pasta dinners the night before a meet or a race. My husband was a swimmer in college, and so in college he would eat pasta and he would eat so much spaghetti, and I was like, where is it all going? Well, what is happening then is you eat that starch, your body takes it and it stores it temporarily as glycogen. Okay, glycogen is stored in your liver for 24 to 48 hours. If you have a big pasta dinner and then the next day you're going to run a marathon, or you're going to swim, or you're going to go do some sort of physical activity, you're going to need a ready energy source at the point in time that your body starts to flag because it's used up all of its readily available glucose. So you'll hear people talk about a second wind, and generally that second wind is the body is getting signals to release glucose from the glycogen stored in the liver into the blood system, and then you're getting that sugar into your cells to be used in cellular respiration. Okay? Unfortunately, if you are not someone who goes and does a physical activity, like you go and sit on the couch and play Stardew Valley for 24 hours and you don't know where time went, okay? Because that game is a time suck. What ends up happening to that glycogen? Well, it can't be stored in our bodies like that for long term. So eventually our bodies will transfer that glycogen into our cells that will then convert that into a lipid as fat, which is why unless you are physically doing those sports and activities, eating 6 plates of pasta is probably not the best plan, okay? So those are going to be your polysaccharides that function as energy storage molecules. Now, some polysaccharides do function as structural components, and while these are still polar compounds, they aren't going to be as hydrophilic or as water-loving. They are going to have some usually additional component parts that do allow them to repel water to some extent. Okay, and this is because I don't want my structural component just dissolving in water, right? If I'm building a sand castle and it gets hit by a wave, it's going to be broken down very quickly. Those walls are not going to last. So we're going to see things like cellulose and chitin playing a role in structural components. And again, they usually have some additional elements or some additional components to them. But they are based in polysaccharides, which means that the basis of plants, the thing that makes plants like trees able to stand up tall, is a sugar. Right? Like, isn't that cool? Or the thing that makes crab and lobster shells so hard to crack is a sugar. It's fascinating. So the reason that that works that way, okay, is because they do not have this kind of wavy branching that we see in starch and glycogen. Instead, these are going to have building block sort of structures that are held together by hydrogen bonds and strengthened by these alternating bond structures, okay? So something like cellulose in a plant cell wall, when you have celery and it's crunchy and stringy and chewy, it's structurally different than what we would see in a potato, which is why potatoes are soft once you boil them. Celery can get soft once you boil it, but you're still breaking it down. You're doing some stuff to these sort of bonds here. However, if you eat raw celery, it's going to have that really strong fibrous feel to it because it has cellulose. Now, you as an animal are not capable of breaking down cellulose on your own. You do not have enzymes that break down cellulose. However, okay, cellulose is still a very important part of our daily diet. You need it as fiber, okay? And again, we know that chitin in the crabs and lobsters and insects isn't breaking down by water, because that would be weird, right? They have an additional layer of lipids over them, which is why they don't break down in water. We're going to talk about lipids next. Okay, so lipids. Lipids are fats. Generally speaking, we go lipids are fats and oils. Awesome. Lipids are so much more than that. They also include waxes and steroids like hormones. They are incredibly versatile and diverse. And in fact, lipids do not have a true monomer or polymer because of the diversity. Instead, what we know about lipids is that they are still made of carbon, hydrogen, and oxygen, just like what we see in our carbohydrates. But lipids have different amounts of carbon, hydrogen, and oxygen, which results in lipids being non-polar. Okay, non-polar molecules do not interact with water. They are hydrophobic because water interacts with things that have charges, and non-polar molecules do not have charges. So as such, lipids are insoluble in water. This is why oil and water don't mix. Right? Isn't that great? We're going to talk through a couple of different things that are used, that are lipids that are used in the cells in the body. And I'm going to identify the ways that these lipids are very different. So when you're filling out the table, you could say that there is no true monomer or polymer, and that's completely acceptable because they are incredibly diverse in their structures and therefore in their functions. Okay, so let's first talk about fatty acids. These are the lipids most people think of. If I ask you to think of a lipid, you probably thought of oils or butters or fats. Okay, and yes, these are going to be incredibly important. Our body needs fatty acids to function effectively. Your brain needs fatty acids to work appropriately, as well as glucose, so that you can actually fire on all cylinders. You need fats in your joints to help keep your joints lubricated. Having fats can keep you warm. Okay, I'm not saying we necessarily need that here in North Carolina, but think about the Arctic and some of the animals that are up there. They have that blubber, which is a thick layer of a kind of saturated fat. Okay, there are two kinds of fats that we're going to talk about that are fatty acids. We're going to talk about unsaturated fatty acids and saturated fatty acids, or oils and animal fats. Okay, so if it is something that is liquid at room temperature, it is a plant fat, and plant fats are called oils. This could be your corn oil, avocado oil, whatever you want, okay? Coconut oil is an unsaturated fat, but it's a little bit special because it's tropical, and so it's not really liquid at room temperature, but that's why it has such a low melting point. Coconut oil melts very, very easily, okay? The reason that unsaturated fats are liquid at room temperature is because when we look at the hydrocarbon chain, okay, and again, there are oxygens in our lipids. We have carbon, hydrogen, and oxygen. Oxygen is just barely represented with these long hydrocarbon chains. There is a double bond between carbons in that chain that results in a kink. This kink does not allow these fats to become solid at room temperature because they slip and slide over one another. And yes, I know how that sounds, okay? Liquids, plant oils are kinky and therefore they're liquid at room temperature. Okay, saturated fats don't have that double bond, and so they're solid at room temperature. And this is your animal fats, your lard, your butter, your bacon fat, your greases that you think about, and even your own fat. Okay, these are going to be saturated. It also means that these are harder to break down because there isn't already an area that you can kind of snip because there's a bend. These are a lot more stable and they can stack up against each other really nicely, which results then in them not being as good for you. Okay, everything in moderation. Don't get me wrong. I am a sucker for butter. Toast is just a butter delivery system. But you need to then make sure that you're balancing that out and maybe not cooking in bacon fat that day, okay? In addition, your body does need some particular animal fats, especially things that are high in omega-3 fatty acids like salmon, or you get that from things like almonds, and that's really good for your brain and your joints and your skin and such. We need these fatty acids. And again, they are a good energy source, but they are harder to break down, which is why it's harder to lose fat than it is to lose muscle, because your body has a harder time breaking these down because it's like, we're saving this for a rainy day, okay? When we look at fatty acids, we can also then look at phospholipids. And phospholipids are very similar to fatty acids in that we have a long hydrocarbon chain. And sometimes that chain is going to have that double bond that results in the kink. But what makes phospholipids different is they have this phosphate group on the top that makes it a polar head with non-polar tails. So overall, your cell lipids, your phospholipids, are still hydrophobic, but they have a hydrophilic part. Okay, they have a polar part and a non-polar part. This is really important. All cells have a cell membrane. The cell membrane is responsible for generating the inside versus the outside of the cell and is used to keep and maintain homeostasis. All cells have this physical structure. It is part of the cell theory. It is part of what we're going to talk about in detail, especially in chapter 5. But we need these phospholipids to function the way that they do to maintain balance of our cells. As such, they are found in literally all cells, bacteria to blue whales, okay? And they are always of this sort of structure, except for archaea. They're a little weird, okay? They're a different kind of group. But, okay, we always see them in this polar head and non-polar tail. And if you were to drop these into water, they will always form this kind of phospholipid bilayer where the non-polar tails are avoiding the water by being together, and it pushes the water either inside or outside of the cell, okay? Last but not least, and not last at all within the lipids themselves, but the last one I want to talk about today is your steroids. Steroids are also often called hormones, and hormones are a key part of maintaining functionality within organ systems. Steroids are often put into the bloodstream to then move around the body to the different locations that they will be needed at. As such, they cannot dissolve in water. Because again, if I have these steroids dissolve in water, my blood is mostly water. That means that they won't get to where they need to be, and so they won't have the functionality that they need to have. But steroids are very, very different in their structure from our phospholipids or our fatty acids. They still have carbon, hydrogen, and oxygen, but now we see 4 fused rings with different functional groups hanging off of those rings at different points. So, the classic example of these steroids are going to be your cholesterol, which is a precursor for both estradiol, which can become estrogen, and testosterone, those hormones that are responsible for secondary sex characteristics. Okay? What I hope you notice is that really cholesterol is not that different from estrogen, is not that different from testosterone. And when we look at estradiol and testosterone, there are very few changes in that estradiol has double bonds in this first functional group along with a hydroxyl group, whereas testosterone has a double bonded oxygen and then a CH3 group hanging off here. Okay? That's the difference. It's also why sometimes we see things like female lions developing manes. Their testosterone levels might be higher, or their cells might be more sensitive to testosterone. And since the estradiol really isn't that different, the cells might pick it up and be like, oh, cool, time to be testosterone. And then they'll start growing manes. So these hormones are responsible for secondary sex characteristics in organisms that are sexually dimorphic, like lions or like humans. And so we might see a variation of those traits being displayed depending on how that organism receives those particular hormones. It's also why anabolic steroids can result, and overuse of those anabolic steroids can result in secondary sex characteristics of the opposite sex. So if a male is using a lot of anabolic steroids, he's basically overwhelming his system, and so his cells, which can receive testosterone, are, but so is the estradiol on those cells, because all individuals produce both of these hormones. You need them for different processes, and so they'll start to develop like no facial hair growth. They'll start to develop female breasts. They might start to have testes shrinkage. And then females are the opposite. They might get the deeper voice. They might develop facial hair. And that's because we are basically flooding our system with artificial hormones. And our cells are just like, okay, cool. I'm going to use what's here. And they start using those in ways that we were not anticipating. Okay? So that is our carbohydrates and our lipids, both which are carbon, hydrogen, and oxygen, but in very different ways. So make sure you can fill in the table for carbs and lipids, and then make sure that you can answer these questions for the check your understanding. Once you've answered these, we will move on to the next part of our lecture. All right, let's talk about proteins and nucleic acids, our last two groups of macromolecules, okay? Both proteins and nucleic acids still have a base of carbon, hydrogen, and oxygen. Remember, that is key to all of these macromolecules because it's important in these organic molecules. However, there are going to be some key differences between proteins and carbohydrates and lipids. First off, proteins are really, really important. While we do know that lipids are important, you can't have a cell membrane without phospholipids, right? And your cells can't function properly, your brain can't function properly without glucose. Proteins are actually going to be the biggest portion of your cell's dry weight. Some estimates say that up to 80% of your cell's dry weight is protein, which means if I took your cells and dried them out and weighed it, a lot of that weight would come from these proteins. And that's because proteins do support, they do metabolism, they do transport, defense, regulation, motion. You cannot move without proteins, your nails, your hair, all of the things, okay? But when we think about proteins, generally people think about meat and nuts and beans and eggs, and that's fantastic. Those are great examples of proteins, okay? All proteins are composed of 20 amino acid monomers, and those monomers are put together in different ways, very much like our alphabet, where we can have 26 letters that write, all of the words that we know in lots of different languages. Proteins do the same sort of thing. So while there's only 20 amino acids, the arrangement of those 20 amino acids allows for proteins to be so diverse and functional, okay? Proteins are different from carbs, carbs and lipids because while they have carbon, hydrogen, and oxygen, we also now have nitrogen involved, okay? So amino acids are going to have a nitrogen, and that nitrogen is what's bonded to a carbon. So as you're linking together your amino acids through that dehydration synthesis reaction, you are taking off a hydrogen of the nitrogen, you are taking off an hydroxyl group from the carbon, and you are forming something called a peptide bond. That is a very specific bond between carbon and nitrogen, and that is what we see in these amino acids, okay? So these monomers are going to be put together, and it's going to form a polypeptide, or a chain of many amino acids linked together. That is your polymer of proteins. A polypeptide is not a full protein until it has been folded. Once that protein is folded into particular ways, it's going to have an active job and duty, and we will talk about that in terms of things like enzymes in Chapter 5 a little bit more in detail. So keep in mind that now we've talked about proteins. We have the amino acids as your monomer, your polypeptide as your polymer, carbon, hydrogen, oxygen, and nitrogen as your elements, and then tons of examples. Very, very key here is that nitrogen. One of the things that does help me remember nitrogen in my proteins is cats. If you have cats, if you've been around cats, if you've been to the zoo and been near big cats, you know that their urine is incredibly potent. It has a very, very strong ammonia smell. That's because cats are obligate carnivores and they only eat meat. That meat is then obviously full of amino acids, and that nitrogen has to get processed and removed as waste, and it is removed in that urine, which is why it has that really strong smell. Okay? I don't know, maybe it helps you, maybe it doesn't, but it helps me. Last but not least of our macromolecules, and probably the macro that no one would ever track, is your nucleic acids, okay? DNA and RNA are in every living thing. Genetic material is in every living thing. If I'm tracking my dietary needs, I am not looking and saying, okay, but how much DNA am I getting from this strawberry that I'm going to eat? However, when we're talking about biology, we cannot ignore nucleic acids. They are really important because they are our information storage molecules, okay? This includes both DNA, deoxyribonucleic acid, and RNA. RNA helps make proteins from the DNA blueprint in living organisms. And the thing that makes nucleic acids different from the other three is that while it has carbon, hydrogen, and oxygen, it also has nitrogen, and we add a phosphate. So we are on to our last added element here. And if you ever watch the Amoeba Sisters, they talk about it as cho-cho-chan-champ, which can be very fun to practice saying. Okay, nucleic acids are essential. We don't build proteins without them. Honestly, nothing else in our cells can function without them. You don't exist without them. Everything that is what you are is based in your DNA. There are nucleotide monomers for the polymer of nucleic acid. So nucleic acids is also its polymer. The monomer is your nucleotides, and there are 5 different nucleotides. Both DNA and RNA have adenine, guanine, and cytosine, with DNA having thymine and RNA having uracil. So the thymine in DNA is changed out for a uracil in RNA. Okay? When we talk about our examples then, DNA and RNA are great, and let's talk about those two things just a little bit more. So DNA has a deoxyribose sugar, and it is a double helix. Guanine pairs with cytosine and adenine pairs with thymine, and between these pairs we have hydrogen bonds. The hydrogen bonds play a role in allowing that DNA to stay zipped and then be unzipped when we need to read the genetic information that is stored in that sequence of bases. We'll get into that a little bit more in chapter 11 when we do protein synthesis. RNA is single-stranded. It has a ribose sugar, and instead of thymine, it has uracil. And RNA is doing the job of carrying the genetic information elsewhere to the cells so that proteins can be made from that information. So proteins and nucleic acids are closely linked. They have a relationship. Your DNA determines the sequence of the amino acids in a protein. And so that sequence is determining what that protein does. And so the DNA being accurately read and used to build the proteins that, as we talked about, do a lot of stuff within your cells in your body is really important. When we have errors in DNA, we can see errors in the proteins that are made. For instance, a classic example being sickle cell hemoglobin or sickle cell anemia. If you have normal hemoglobin, you have an amino acid called glutamine. In sickle cell hemoglobin, that glutamine is changed out for a valine in that amino acid sequence. It's the difference of saying cart and can't. Okay, when that valine is changed out, it means the protein doesn't have the same meaning anymore, and so it doesn't fold properly, and therefore it doesn't do its job appropriately, which can be very problematic. And we'll talk more about mutations and what happens with those mutations in both genetics and protein synthesis later this semester. Make sure that you can fill out your table for proteins and nucleic acids and answer these understanding questions. As always, if you have questions, please let me know. Thank you and have a good day.